Imagination is more important than knowledge. Knowledge is limited. Imagination encircles the world, said Albert Einstein. The world is changed by those who are able to see beyond the current limits of knowledge, beyond the current levels of understanding of the world we are living in. Those people who are able to change the world can use their imagination and come up with something totally new, with new original ideas. And that's what musicians and scientists have in common. They pave new paths that help us to think about the world we are living in a different way, and that help us to understand the world we are living in a better way, so we can improve or even just to survive. Tonight you will meet 3 distinguished scientists, 3 Nobel Prize laureates who were able to change the world, to come up not only with the idea using the imagination, not only using the knowledge to make sure that they get the result, but they were also able to work so hard, use the perseverance and dedication to truly make the world happen, to change the world we are living in. Tonight, we will go into the fields of chemistry, immunology, as well as we will talk about quantum mechanics, the next revolution we are experiencing right now and in the near future. Tonight, I have the honor to welcome you to the dialogue between music and science. It's an honor to welcome you to Sounds of Science 2024. Good evening to you all. We are at, and I do believe you would agree, in a magnificent, truly magnificent estate theater that was founded in 1783. And this is actually the very last place, which is still being used as a theater, where Wolfgang Amadeus Mozart played. The very last in the world. It was actually Don Giovanni that was played here in this very theater. For the opening speech, I would like to kindly ask His Excellency, Mr. Ambassador, Monsieur Stéphane Cruzon, to join me on stage. Ambassador of France to the Czech Republic. Your Excellency.
Merci beaucoup, bonsoir, vážení hosté, dámy a pánové. Je mi velkým potěšením přivítat vás v tomto krásném divadle, které je plné hudebné historie. Dnes večer se již potřetí v rámci akce Sounds of Science spojí věda a umění. Dnes večer tu máme 3 francouzské nositele Nobelové ceny a 3 renomované české hudebníky. And Sounds of Science is honoring the long history of successful relationship between science and the arts, each of them influencing and nurturing the other. This edition is also taking place on a very special occasion, the 30th anniversary of the French Embassy's scientific prizes. Every year, we give prizes to young Czech scientists in fields as varied as chemistry, medicine, nuclear science, pharmacy, computer science, environmental science, and social sciences, and the humanities. And today, this morning, at the French Embassy in the Palais Bucquois, we gave our prizes to so many young Czech talented scientists. And we're very proud of the longevity of this program, thanks to the continued support of our partners and sponsors. And we are also so honored and so happy to have here 3 fantastic Nobel Prize winners, Professor Alain, Professor Jules Hoffmann. Thank you for your enthusiastic response to our call, as well as Ivo Kahanek, Mihai Rataj, and Jan Trojan for accepting the challenge of connecting both worlds and taking us on a very special musical journey. And finally, Professor Jean-Marie Lehn, not only you came up first with the idea of the Sounds of Science conference, but it also you have provided constant support throughout these years to promote young French scientists, and we are extremely grateful for that. Merci beaucoup.
Thank you very much. Merci beaucoup. It will be truly a treat, not only concerning the science, but also concerning the music, because we'll be traveling in time. We'll be starting in history of music, and then we will close up this very evening with the future of music using AI, the combination of the past and the future. And all also thanks to the support of the scientific partners of the whole event, Czech Academy of Scientists, Charles University, University of Chemistry and Technology Prague, as well as Faculty of Nuclear Sciences and Physical Engineering of Czech Technical University in Prague. And that's why I would like to kindly ask Professor Eva Zažimalová, the president of the Czech Academy of Sciences, to join me on stage. Professor, thank you very much for joining us. Good evening too.
Your Excellency, Mr. Ambassador, dear distinguished Nobel Prize laureates, dear guests, ladies and gentlemen, I am very pleased and it's an honor for me to greet you on behalf of the Czech Academy of Sciences to today's Sounds of Science. Sounds of Science is already a traditional event, but it is much more than a series of presentations and musical performances. It is a celebration of curiosity and never-ending research. The Sounds of Science goes across many scientific disciplines and reminds us of power and beauty of science. While science helps us to understand the world intellectually, music does it emotionally. In the following moments, we are going to listen leading scientists and visionaries which push forward boundaries of what we know and what we can afford. And they are also pointed to the fact that every and any scientific discovery, even the very small one, always contributes a note and a tone to the great symphony and profound symphony of science. I wish you a very nice evening and thank you.
Thank you very much, Professor Zažimalová, the president of Czech Academy of Sciences. Now I would like to kindly ask the rector, the president of the oldest university in Central Europe and the largest one in the Czech Republic, Mrs. Milena Králíčková, rector of the Charles University. Professor, thank you very much for joining us.
Your Excellency, Mr. Ambassador, dear professors, dear guests, ladies and gentlemen, it's my great pleasure and real honor to greet you and welcome you on behalf of Czech Rectors Conference and Charles University on this occasion of Sound of Science 2024. It was said already that this is the place, these walls remember Wolfgang Amadeus Mozart, and right next to this building there is Carolinum, which for almost 700 years remembers scientists, not only Jan Hus, but also 3 Nobel Prize winners. I would like to mention that graduates from Charles University were, for example, Karl Corrie and Gerta Corrie, both of them Nobel Prize winners for medicine, and she was actually the first woman in history that was awarded for Nobel Prize for medicine. Even these 2 buildings show how close arts and science have. So I would like to thank all of you who are behind organizing Sounds of Science. I would like to thank Mr. Ambassador and all the partners that prepared such amazing evening, and I just wish you enjoy Sounds of Science and let us all fly on the wings of arts and science. Thank you.
Thank you very much indeed. Professor Milena Králíčková, Rector of Charles University. She has mentioned the partners. Thanks to the partners we can meet here. Among the partners there are Institute of Physics of the Czech Academy of Sciences, Institute of Organic Chemistry and Biochemistry Tech Foundation, BNP Paribas, EDF, L'Oréal Czech Republic, and Young Talents Foundation. The first man who will join us on the stage to talk about science is a man who said, quote, "I wanted to do more literature, history, philosophy, languages. Insects? That happened mainly because of my father. Without my father, I would not have done science at all. He has done science. And he's the Nobel Prize laureate in physiology or medicine. He worked on fruit flies, and thanks to his groundbreaking work on fruit flies, he was able to determine one of the crucial lines of defense of our bodies, the innate immunity. The Nobel Prize laureate himself, Jules Hoffmann."
Ladies and gentlemen, Mr. Ambassador, ladies and gentlemen, dear colleagues and friends. So what I would like to do we have 20 minutes. What I would like to do with you this evening, take you through the efforts which we have developed to get some results which are of some relevance for basic science and for applied science. If I may have the first slide. So let me take you through this rapidly. This is life expectancy during history. Life expectancy means the years which pass from birth to death. And we have values for minus 10,000 years, for minus 3,000 years. There was no big change. And then Breslau here, minus 1,700 years. Then we're getting closer to Liverpool. And you see there's no change. The life expectancy, the mean value, which would be the red line here, is around 25 years. Even in Napoleonic wars, it was around that value. And then, all of a sudden, we have a value here from the UK. It would be the same here in Czech Republic. And you see that there's a fantastic gap. All of a sudden, life expectancy has progressed dramatically. So this is actually the basic questions which we want to address together this evening, shortly. And let me say, to make things a little bit clearer, I will go by steps. Step 1, step up to step 7. So step 1 is, in the mid-late 19th century, scientific discoveries led to understand the microbial origins of infections. It's infections which explain this terrible gap between the years of the 19th century and then finally in the 20th century. Now, infections were believed to be the result of bad behavior of people, and priests would tell you that you had done or your country had done mistakes and so on. You would have processing and prayers and to overcome that. And this then showed us that 2 people here showed us that, in fact, it was not divine powers, but it was just germs which produced this change in longevity. This is Louis Pasteur in Paris at that time, of course, and Robert Koch in Germany. Robert Koch became a Nobel laureate. Pasteur did not, because he died before the Nobel Prize was created in 1901. And so those are really the 2 pioneers who showed that infection is one of the main, it's not the only, but one of the main causes of reduced longevity. And then, rapidly after that, 10 years ago, immunological studies became really real. And Alain Metchnikov, who was of Russian origin, and who worked in the end of his life in the Pasteur Institute in Paris, and Paul Ehrlich were the people who really addressed the question. So, okay, there are microbes, but does the body recognize them? Can it react against those? And Metchnikov discovered phagocytosis, and phagocytosis means taking up a microbe or any other thing and destroying it. And he created the concept of innate immunity. On the other hand, Paul Ehrlich in Germany, he discovered a serum-based immunity, which is an immunity. They showed that if you injected several toxins into animals, you would induce a specific serum-based immunity, which had some specificity. In contrast to Metchnikov, this was, of course, serum is devoid of blood cells. And for phagocytosis, which was due to blood cells, in the work of Metchnikov, could not account for this. As in the work of Metchnikov, there was no specificity, whereas in the work of Ehrlich, there was a very intense specificity. So this led Ehrlich to propose a concept of adaptive immunity. That was a crucial moment in the evolution of immunity, in the start of immunity. and which has persisted until now. And now, the two shared the Nobel Prize in 1901, although they defended totally different views. And instead of being very happy about this, they were angry each at the fact that the other also got a share of the Nobel Prize. To such an extent that Metchnikoff did not go to Stockholm to collect the prize, and they published in his lecture, in his Nobel lecture, Ehrlich never mentioned the name of Metchnikoff. Well, that was in former times. You know, science has evolved, and now the ego is not there anymore, and now people collaborate. Okay, people collaborate. When I say this sentence, I like to say it. Always, I have the same reaction from the people in the room. Say, oh yes. So they believe that we still have a strong ego. Okay, then let's continue now, and we go to step 3. And in step 3, so what I want to summarize, we don't have much time, so I will be there were, after those pioneers, there was the work of Bordet, who found the complement, the work of Landsteiner, who found the blood groups, and then the work of the people who discovered the antibiotics. And all this stayed still in a certain area of understanding between innate and adaptive immunity until the work of Gerald Edelman and Rodney Portner, who then established the chemical nature of antibodies from the blood, from the plasma, not the serum. And so they really started a new view of the immune system. And that was the chemical nature of antibodies was the beginning of really the domination, total domination of adaptive immunity. Innate immunity was considered as, well, you could play around with it and insects and so on, but it was not really relevant. They shared the Nobel Prize in 1972. And they opened this new, as I just mentioned, new phase of research. From that time on, nearly most of the studies were on adaptive immunity. And now I will start speaking about our work in Strasbourg. And this is the Institute of Zoology in Strasbourg, where I worked. And you recognize the style of the building. It's relatively Germanic. At that time, Strasbourg was part of Germany. And my supervisor was Professor Pierre Jules. And just this is interesting to point out, he had worked his full career, 30 years before we met, he had worked on endocrine control of reproduction of insects, and essentially had worked on grasshoppers, Locusta migratory. At that time, France had some, how should I call that, some responsibilities in North Africa. So they knew that this could be a way to control the hosts of grasshoppers. And then Professor Joly, it's exactly, was 60 years ago. He said to me, well, look, I have worked for 30 years on this, and we have sort of transplanted from one individual to another all the putative endocrine glands. So this comes to an end. There was no biochemistry available at that time, at least in a laboratory like that of Pierre Joly. And he said, but strangely enough, I never took any precautions of asepsis, and there was never development, I noticed never development of an infection. So I propose that insects are very resistant to infections. Would you like to take this up for your thesis work? Initially I was delighted, and then after that I noticed I had no idea what immunology was like, and worse, Monsieur Joly had no idea either. And at that time, immunology was taught, at least in our universities, in medical school. But we were in the zoology department in the Faculty of Sciences, so it was a tough start. Until we changed from Locusta to flies, and eventually to Drosophila, as you see in the slide, and you see the difference in size of these animals. Now, step number 4, we continue now what I said. So the first thing which I must say, very frankly, it took us years. to get this work started. We had only like microscopy as a new tool, and the rest was histology and so on. And then we came to find there were inducible peptides, and you see here this is a big fly, and this fly we identified various. We're not going through the names as we have no time, and they do not really matter for what the purpose of this evening. So we saw that they produced a series of peptides which were active against at that time was bacteria and fungi. At the same time, Hans Bohmann, whom I admired very much in Stockholm, he worked on butterflies, and he discovered cecropins and adhesins, also antimicrobial peptides. And then, shortly after that, two of our colleagues in the United States at UCLA, Tom Gens and Bob Lehrer, found that in humans you had also similar molecules which they referred to as alpha-beta defensins, cathelicidins, drosomycin, like the one name we had given in the fly. And in the fly, the peptides are produced in the equivalent of a liver, which would be the fat body, whereas in humans it is essentially in the mouth, in the lung, in the intestine, urine tract, reproductive tract, and blood cells and skin. So it's a berry. Okay. So then started a period. This is step 5. We're getting close to the end, not fully yet. And no, we haven't done most of the work yet, but that will be shorter. So I just want you to understand how things went. And it was very difficult to get funding for this type of study. The immunologists were not interested in, the true immunologists were not interested in what we were doing. And even CNRS, our research institution in France, said, well, come on, this is not really immunity, this is just antibacterial stuff. And in insects, and you should stop doing this. And they even wanted, when my supervisor was retiring, they wanted us to stop working at the university and go into agricultural research. But we were fighting. And now, what I show here is that we, over the years, it took nearly 25 years, we succeeded in deciphering the whole cascade, which goes from recognition of the aggressor, in this case a fungus, to building up a defense. In this case, what I show is, this is a famous fly which made the cover of the journal Cell, and you see a fly which is deficient for a pathway, which I'm going to illustrate in a moment. It's unable to fight the fungus which is coming out of every part of the body. Okay, now, what we then did, we started working over the years, again, from the ligand which induces this immune reaction. I say now immune reaction because now it's accepted, via circulating receptors in the blood, going to the cleavage of a circulating cytokine, which goes by the name of Spätzle, and then a transmembrane receptor toll, and finally a nuclear transactivator, which goes by the name of factor kappa B, which was known to be present in B cells of humans. And this, in the end, went to produce this drosomycin, which I've mentioned already, which is a natural antibiotic. So the dots in the arrows indicate they are intermediate molecules, and finally, we succeed in, and occasionally with the input of other people on other systems, we succeed in determining the whole pathway. There's more than 20 molecules involved here, from the ligand to the efficient defense. And everything had been identified then. So this was actually the most interesting result of our studies. These are two of the essential work. We had a large laboratory. The laboratory is still large, but I'm not in that laboratory anymore. And Bruno Lemaitre and Jean-Marc Reichard did the end phase of this work. And so after this, I propose that we go to the next step. And then we make the joint, we will establish a bridge between innate immunity. What I've shown so far is all innate immunity, no memory. we go to the we bridge over to adaptive immunity. This is Ralph Steinman, who shared the Nobel Prize with Bruce Beutler and me in 2011. And what he had been working on dendritic cells, which you see here, they are present in lymph nodes and so on. And what he had shown was that microbial ligands, microbial fragments, getting into the cell either directly via phagocytosis or through followed by the demolishment of the microbes inside the cell. Those two systems activated NF-kappa B, which is a transactivator. I suppose we all know transactivators are proteins which activate the transcription of a gene in any biological system. And NF-kappa B is one of the central transactivators of immune reactions in general. We know now both in insects and in mammals. So what then happens is that we activate, well, we have the innate immune response, e.g. the antimicrobial peptides, which we have seen together a few minutes ago, and also the activation of the adaptive immune response by innate immunity. That is to say, now, in historical terms, we would say Metchnikoff activates Ehrlich here. And so it was a good idea to share the Nobel Prize between the two. And so this then will transform the naive lymphocytes into effector and memory T or B cells, essentially B cells. Now, there are three things happening here. One is the antigen presentation, so everything which is foreign in this cell would be called an antigen, which is bound to a peptide. We have not to go into the detail of that here, so it's called MHC restriction, no relevance to us this afternoon, but big relevance to us if we're sick. And then the second signal, I just put it in because it has become so important in present-day medicine. There's a co-stimulating molecule. There has to be co-stimulation. It's not enough to present the antigen. You have to have a co-stimulator. And those co-stimulators are the ones which are blocked in the system of Honjo and Allison, which I'll show in the last slide. So, and then the third very important, it doesn't want. Why doesn't it want? Okay, because I said it's important, so it shies from coming out. A very important activity is the production of messengers of the immune system, which are referred to as cytokines. And there are many of those cytokines. And in our human body, there are probably more than 100, and chemokines. And so they then have also special roles to play in the defense against microorganisms. Oh, here they come. So cytokines, chemokines, particularly for inflammation. Now, we go to this, we show this now. When these results came out, that was in the early 90s, with Ralph Steinman, we were in contact with Ralph Steinman, and then we all agreed that we would replace innate immune receptors by toll-like receptors. So the receptors which we had found in the system, which had been referred to toll. So I can, if you have questions, I can explain why we use that term of toll. Now, let me just, in this slide, summarize. Now I'm going to the mammalian system. I'm going to really to mammals like mice and humans. And there are 12 of these toll receptors, or toll-like receptors, if you wish, in mammals, and tolls 9 in flies. Now, what are their functional involvements? This is important. We see that they play a role in infections. That's the way they were found. They also play a role in inflammation, still the way they were found, and in autoimmunity, and in vaccination, that's where they play a very big role, namely in COVID recently, as you have learned, in allergen recognition, in cancer, that was totally unexpected for us, and then in central nervous system, I come back to those points a little bit later, in the kidney and in the cardiovascular system. So what this tells us is that this type of receptors is expressed in the majority of tissues, and this is in humans, the majority of tissues, so we can anticipate that plays a role. And that's one of the important topics of research nowadays. Let me rapidly go to the fly. No, this computer doesn't like the flies. Okay, here they come. So here we're told in blue, involved in development, which had been shown by Nusselt and Follard, defense peptides also, and in inflammation. So this is a little bit banal now, but I mean for this audience, but what is interesting is to see that since we published our paper in '96, in which we showed from the beginning of the infection to the result of the infection and to the defense, there have been some 70,000 papers which were published on TOLs and TLRs over the whole scientific community. So this has become a very important topic in physiology. And let me shortly go to step 8 and show, I mentioned that I grew up as a zoologist and in the Institute of Zoology, and now innate immunity is shown here. Innate immunity is present in invertebrates, which make up 95% of extant species nowadays still. 95% of all animals on Earth, biodiversity, are invertebrates. Only 5% are vertebrates, and I'll illustrate one of those here. The important point is that they make up only 5% of all species on Earth, and they have both innate and adaptive immunity. That's the important point here. And as I've shown now with Ralph Steinman, innate immunity gives a signal to adaptive immunity. So that's to say the whole system is not as our predecessors had thought. It's a binary system. It's a complementary system. Innate, first line of defense, sees the invader coming, fights it through phagocytosis, and then gives a signal to adaptive immunity, and adaptive immunity will then do the rest. Now, let me rapidly go to the end now, but my time is soon over. The period of the 60s and 70s witnessed the identification of the lymphocytes. So up to that year, you realize that was after I took up my topic, the cells of the adaptive immune system in vertebrates had not been identified yet. And then they were, through the work of a whole series of colleagues, Max Cooper in the US, Jack Mill in Australia, James Govans in the UK. And as a result of this, two very well-known persons, James Ellison in the US and Honjo Tazuku in Japan, developed a system which we refer to as checkpoint control. That is to say, the second signal, the co-activation, which I've shown in the Steinman slide, the co-activation is blocked, and then the response of lymphocytes cannot be as dramatic as normally would occur. And so in the case of cancer cells, then you can fight cancer cells, and but the drawback is that occasionally if you are too efficient, there will be autoimmunity coming up because the lymphocytes become too aggressive. And now I will just, this is my last slide, and I'm in the time limit, and I want to wrap all this up. After so many years being involved happily in all these stories, and partially involved, of course, because there are thousands of people having worked on this. In our lab, we were variously between 50 and 100. And the present-day understanding is marked here. So the immune system is a powerful global defense mechanism, global defense, which is activated by recognition and sensing of any changes in homeostasis in an organism. So that can be in a cancer cell, can be in a nervous cell, it can be in a blood cell, any of those. And so I'm showing this by microbial aggressors. That's how the system was initially shown to exist, and Metchnikoff, Ehrlich, and so on. That's a classical acceptances. And then internal modifications. This is new, this is recent. Because even until 2020, until that time we worked essentially in a laboratory on flies, when I asked colleagues from distinguished mammalian immunologists, when they were working on cancer, wouldn't you expect that cancer cells would also be recognized in mice by the immune system? They said, no way, because the immune system has evolved to distinguish self from non-self. And the cancer cells are self, of course. They're a little bit changed. And now we know that this was a short view, because now, as I put here, any changes. Now, in a cancer cell, you have production of a whole series of new molecules, which we call, or people call, neoantigens. And they represent a change in homeostasis. And this internal modification will then induce the immune system. So the immune system is also trained to attack the cancer cells. And what is more, age-related effects, which can be in the most spectacular or horrible situation, can be neurodegeneration. It also has been shown recently. This is just a few years ago. It was not expected. It has been shown to be modifications are provocative of the immune system, which will react to this system. How it does and how it succeeds is not yet known, but I can state that maybe 200 or 300 of the best laboratories in the world are now exploring the role of the immune system in cancer and in neurodegeneration. And also many companies are working in that direction. And so with that, I will finish my presentation. I will thank you for your attention. And I wanted to make, as we like to do in these type of meetings, the way we got through basic research without knowing anything. Monsieur Joly and myself, we didn't know anything about immunology. How we could participate in the evolution of the knowledge here. And we're in a totally different situation nowadays from what was believed even 20 years ago. Thank you very much for your attention.
You could have heard the cheers during your presentation, even from the outside. The basketball fans were impressed.
Okay, you're very kind.
How many fruit flies have you worked with?
Well, 18,214.
Excuse me?
I forgot, 25,800. No, I mean, we never, that's one of the advantages of this type of model. You don't count the number of animals which you kill.
Yeah.
Okay,
that was not the point of my question, honestly. It's escalating quite quickly, honestly. That's what I did not expect it, because I wanted to talk about what you have mentioned at the end of your talk, the improvement of technology, the improvement of science. Because when you were identifying the one single peptide, you needed to immunize 100,000 fruit flies individually to make sure that you can identify it.
Well, initially we did this on the big flies, which you saw on my slide. And there we extracted about 1 liter of blood. It was my wife who was in the audience who did that work to identify diphtheria. So it was 1 liter of blood from a fly, even a big fly. It's quite intense work, yes.
How was that? Pardon? please don't hesitate to join us. Please welcome on stage, wife of Professor Hoffmann. Yeah, please don't hesitate to join us. Absolutely. Please, Mrs. Hoffmann. How was that to work with him, to work with 1 liter of blood with flies? It didn't happen in one day. Good to know.
And
Yeah.
I was not alone.
You were not alone doing it all by yourself. But did you enjoy the walk with him, to work with him?
Yes, very much.
Very much.
Yeah.
How come? Why? What was the main reason? Why did you like to work with him?
Don't become
He
too
was a good
personal.
boss.
He was a good boss, okay.
He listened to my counsels
Oh, he listened to your counsels, okay.
sometimes.
And he still does, I hope.
And I hope so too.
You hope so. Thank you very much indeed. Ms. Hoffmann, Jules Hoffmann, thank you very much for joining us.
Okay.
Thank you very much for being part of Sounds of Science 2024. Our next speaker, the second Nobel Prize laureate who will join us on stage, says it all in one sentence. He says, quote, "Science shapes the future of humanity." And he is right. And he stresses the importance of chemistry. He says, "Chemistry penetrates our life in practical terms even more than any other form of human culture." That's what especially physicists like to hear. He believes that the periodic table, the playground for chemists, is the most fantastic product. He has been coming to the Czech Republic since Velvet Revolution very regularly, we may say. He's the one who's coming to inspire, to talk to general public as well as to inspire a new generation of scientists. It was the year 1962 when he arrived to Czechoslovakia for the very first time, when he was the PhD student. He is the one who described the process that helps molecules identify one another. He is the father of supramolecular chemistry. Ladies and gentlemen, Jean-Marie Lehn. Jean-Marie, thank you very much for joining us.
Good to hear.
Welcome on stage.
You know too much.
You are too kind. Thank you very much.
Okay, well, back here in Prague, and then with Danielle, I have met several years ago. And I will try to position chemistry within the sciences, but what is important is the science, not just chemistry or whatever. So, this you know, we are there. And this is very important. That's why I'm so happy to be here. To be able to say Mozart was standing somewhere in this room. This is quite incredible. Don't you think so? It's incredible to have the first chord of the ouverture of Don Giovanni. Anyway, that's the past, but music continues and science continues. So I would like just to show you a few steps towards life and thought, because thought is the most important, much more important than life. And chemistry, one can say, plays the score. We will start very, very early on. And modern science, cosmology, has now shown us that after initial fluctuation, quantum fluctuation, the universe increased by 10 to the 78 in 10 to the minus 32 seconds. That's really just after the Big Bang. At that time, there was only physics. There was no chemistry. It was much too hot, and the universe cooled down, particles formed, particles got together to make atoms, and then atoms got together to make molecules. And chemistry started around here. Then, by complexification of these entities called molecules, more complex molecules formed. They started to form also sort of containers until primitive cells existed. And on our planet Earth, biology evolved around this time. Of course, there are probably other planets where it did some similar things earlier or later. But the most important feature is to be able to think. That's why we are here. Here is the thinker of Auguste Rodin. these are steps towards life and thought, very broad steps, I must admit, brought by science and music. And this person you know. Close your eyes a moment and just think that Mozart was here. Some molecules may be still floating around. Could well be. Now, what is the big question we have to ask? How does matter become complex? How is it possible for Mozart to exist? Let's look at 3 areas of science. One is physics. Physics deals with the laws of the universe, the basic laws on which everything else depends. That is, how, why, all these questions can be asked. Then, biology can be considered as studying the rules of life. These are not laws, they are rules. The laws are given by the laws of the universe, the laws of physics. Now, what is chemistry doing? Chemistry tries to build the bridge from the general laws to the way they can be used to function. And this is also, in some respects, sometimes I say, the laws of acoustics don't explain all the pieces of music you can derive from it. So there is a way to go from the general laws, and one has to understand how one can go to specific expressions of these laws, like a 2-legged organism on a small planet in the universe having invented science. How can this happen? Because I personally think, and I'm not the only one, that our universe has a property which is it can self-organize on the basis of these laws, and this is in our universe a cosmic imperative. In other words, I would say we cannot avoid organization. It's not an accident, it's a necessity. Now, mankind then looked at things around and tried to see what is things called matter, called stones and so on, are made of. And in the middle of the 19th century, it was clear that there were these bricks which had been discovered, could be put into a table. This table is, of course, well known for all the chemists, all the scientists around in the room. They know this. They say, okay, what is he talking about? What is simple things? Simply because it always fascinates me and that we have this table. Because this table contains the bricks of visible matter in our universe. In our universe, there is dark matter, there is dark energy. Altogether, they make up 95% of the universe. The 5% remaining, this is the matter that matters. It's our matter. And it's made up of 92 natural elements, and everywhere in our universe, these elements are present, and anything that exists in the universe are made, the visible matter, are made of these elements. And that's the sounds of the elements, you may say, the music of chemistry. Now, when atoms go together, the bricks make houses, molecules. Let's look at 2 milestones, just to show, give you an idea about evolution. In 1828, German chemist Böhler made urea in the laboratory. Urea is contained in a living organism, and this showed that you do not need an organism to make something which is contained in the organism. In other words, it's a chemical synthesis, a production of this urea in the laboratory. Many years later, in the 1970s, a much more complicated molecule, vitamin B12, you can see here, which we have in our body, was made in the laboratory by 2 groups, one directed by Robert Burns Woodward at Harvard, and the other one by Albert Eschenmoser at ETH in Zurich. Enormous progress between the two in the art and science of making complicated molecules in the laboratory, and of course it has continued. And now, of course, one has methods for making complicated, complex molecules which are much more advanced than the ones used in the 1970s. Then we can ask a question, beyond this idea of molecular chemistry, making molecules from atoms, houses, buildings, from bricks, what else? I want first to show the relationship also with music, because chemists have also been composers. And Borodin, he considered himself more like a chemist than a composer. Most of you know, of course, Borodin as a composer, but he said, I'm doing music on the weekends. The rest of the time, I'm a chemist. And there are several reactions. He had, of course, many symphonies, quartets, had an opera, and also there are two reactions on his name, two Borodin reactions, which are ways of transforming chemistry, chemical substances into one another. Now, I want also, and Monsieur l'Ambassadeur, you like Wozzeck, as I like it. So I think you should be interested in that. In Wozzeck, Act 1, Scene 4, the doctor speaks to Wozzeck and says, there is a revolution in the sciences. Albumin, fats, carbohydrates, and even oxyaldehyde anhydride. I think that's the only opera where I know that there's a real chemical name. Oxyaldehyde anhydride. That's acetic anhydride, it's called. So I wanted just to bring that because I think we talked at lunch together. And so in Wozzeck, not only is this for us, for Monsieur l'Ambassadeur and for myself, one of the first 3 operas in our list of preferred operas, but even for a chemist, there's another meaning. Now, what beyond molecular chemistry? We heard about these complexities of the immune system. And of course, I could just build on that. Let's just show here a cancer cell, the blue ball, 2 killer cells, the ones in violet, and these killer cells, they should find the cancer cell and destroy it. Similarly, when you have a virus, the virus, when it hits its target, it can infect, here, the HIV virus and red blood cells. What tells the cells that the other one is the partner or the one they should find, try to find, and maybe destroy, or for the virus particle so that it can infect? What happens if something happens between the molecules? All these entities you see, the cancer cell, the killer cells, the white blood cell, the HIV virus, these are chemical buildings which interact with one another, and something happens at the place where they contact. And this is, they must recognize, and though there is therefore a chemical process which exists in assemblies of molecules, which is that they can recognize one another. And this then led to the idea that there is a chemistry beyond molecular chemistry. And in fact, everything in condensed matter is supramolecular chemistry, where first you have to bind things together. If not, they will then ignore each other if they don't bind. And more importantly, there is information. Molecules contain information which is stored in the structure and which is processed by the way in which they interact. And this concept was announced in 1894 by Emil Fischer, when he said, for an enzyme to act on its substrate, they have to fit together like Schloss und Schlüssel, lock and key. And he got his PhD in Strasbourg. At that time, we were part of an empire on the other side of the river Rhine. Now we are in Europe. And I hope you realize what that means. It's our future. Yes. Thank you. I think we are all very sensitive to that, I hope. So chemistry is the science of informed matter. It is this information is stored in the molecules, in their shape, in their structure, in the atoms which are there, and then the processing of that information occurs through contacts, through these entities. So there's molecular storage of information, and one of the most fantastic storage systems is, of course, the genetic information stored in a double helix. Everybody knows about double helices. And this genetic information is written in a very poor way. Just 4 letters. And these letters are very simple chemical groups. There are these simple things here. This this, and this, adenine, guanine, thymine, cytosine, very simple chemicals. And this is enough to make the language of, you might call, life. How is this? Storing it is the sequence, but reading it is by contacts. So the storage is molecular, and the reading, processing is supramolecular because they have to get together, they have to touch each other by 2 points of interaction or 3 points. So you just realize how basic this is, basic meaning even poor in terms of complexity. 4 letters, 2 pairings, and that's enough. Of course, things get a bit more complicated when there are more complicated molecules, but this is the basis. We can say that the music of the genes is just written with 4 notes. A, G, I put the D because there's no T in the music. A, c'est du La, G is Sol, D is Ré, and C is Do. A, G, T, C is the genetics, H, E, D, C, it's the music. And you play it with 2 hands, 2 ways of putting it together. The genes have one thing which is interesting. The name of Bach is a name which can be written with sounds B, A, C, H, H, C. And Bach was very aware of that because the last fugue in the art of the fugue is built on his name. There are not many people who can see that. Now, molecular recognition, therefore, is sort of the art and science of making, let's say, locks for keys or keys for locks. And this then leads to an association, which is called a supramolecular entity, and very many laboratories have been working on that, because it has also a number of potential applications we'll see in a moment, which are, first of all, obviously, drug discovery is to make a key for a biological lock, a key which will find its lock and do something to it, inhibit, activate, do something. And this is, say, the very simple, simplistic way of seeing what is drug discovery, making keys for given locks and having those as selective as possible so as to have no side effects. We also made over the years a compound for medical diagnostics. That's a cage which has a europium ion, again Europe, I like that one, a europium ion which has a red luminescence, and one attaches it to an immunoprotein, and then one can develop an apparatus for medical diagnostics. Gene transfer is also part of that. If you want to go through a cell membrane, you have to catch the entity you want to bring through, to bind to it, and to help it to go into the cell, which then leads to the possibility of doing gene therapy and biotechnology. That means genetically modified organisms, and here I would like to advocate the need for making genetically modified organisms. Finally, biomaterials. Biomaterials can be made by using the normal materials, so to say, and making them biocompatible. And I wanted just to indicate here how complicated and unpredictable, you can predict some things, but often quite difficult to predict, it can be. In 1990, we published the first paper on what is now called supramolecular polymers. 23 years later, in 2013, there was a small company in the Netherlands which had transformed our supramolecular polymers into biopolymers, which are biodegradable and which can be used to make cardiac implants for children having a congenital cardiac malformation. And here is the little Dominika and Leo Bokeria. She was transplanted by Leo Bokeria in the Bakulev Center for Pediatric Surgery in Moscow. She looks happy. He looks happy too. We were happy also. Now, what about self-organization? When a virus forms, a virus is not very living, it's really living. It's a bunch of molecules in an organized fashion. The tobacco mosaic virus, which is one of the first viruses, the first, which was really understood how it assembles, it is made from 2,130 protein subunits, one molecule of the genome. It is an RNA here. And this spontaneously forms this thing, which is looks like a complicated construction. But the bricks contain all the information necessary and all the positioning of the interaction so that two bricks can come together and then spontaneously generate this type of helical tower, which in the middle contains the genome of the virus. It looks like magic, but it's not magic. We understand everything this is doing, but nevertheless, it's quite impressive. And this is sort of a program written in matter, so to say. As I said earlier, the chemistry is the science of informed matter, biology too, of course, and even more complex than chemical ones. And these programmed chemical systems are a sort of way to have perhaps also in the future, and well, hopefully, to go from the need to make, to let the object make itself by making it in such a way, giving it such a structure that spontaneously will recognize others. The tobacco mosaic virus is rather simple because it has 2,000 protein subunits which are identical. They just have the right shape to be able to assemble. Now, this can be considered as the ultimate fabrication, which is of interest in principle. It can become more and more of interest for nanoscience and nanotechnology. And in fact, just to illustrate what I tell you here, one can make this in the laboratory, for instance, by making molecules which can bind metal ions like iron or cobalt or copper or silver or lead and so on. And then when these are well designed, these molecules, in accordance also with the way they interact with the metal ions, which can be in a tetrahedral fashion or an octahedral fashion or a trigonal bipyramidal fashion. In other words, there are rules for putting things together. And then if you know these rules, if you begin to understand them, you can then design objects which will spontaneously lead to a result which you can predict. And here is, for instance, an artificial double helix on the here, or an artificial triple helix. You have to make, of course, the strands, but then you mix it with the correct metal ion and you automatically get this. The same is true for this sort of grid where the three horizontal molecules in blue, the three vertical ones in red, they bind 9 silver atoms in one step. About one step, I don't know, because maybe it's a progressive one, but you see only one step if you don't do it, you don't analyze it by fast processes, fast detections, and you get this grid where metal ions, silvers, are arranged 3 by 3. That means 9 of them. You can make a nanocylinder. Here you have 3 linear molecules, 4 flat ones, and the linear ones, the flat ones, plus 12 metal ions. That means 19 components altogether. They just form, nothing to do. You have to design them, you have to make the pieces, but once you have the pieces, you just mix them, and you get the result. And here are some other ones, which thanks to computers and the way you can make nicely colored objects, pictures, you have them. This is other types. And of course, this is just very simple compared to what one does nowadays in terms of spontaneous assembly of complex entities into even more complex structures. Now, the next step is, and this is something I just point out because that's now what we are mostly working on. Here, the first, what I have illustrated is mostly the self-organization by design. You think out the molecules, the system. This is the information has to be present in the right positions, in the right structure. And this sort of leads to a program which then functions and produces the final object. With selection, that means that you introduce a new parameter, which is that the system can select among a diversity of bricks, can select the ones which are required for building up the final structure. Another property is required here, that is dynamics. In other words, since chemical processes, be they molecular or supramolecular or reactivity, they happen by a collision. And if this collision gives something which is too strongly bound, then you cannot equilibrate. You cannot look for something else, for searching for something which may be better. Better meaning thermodynamically better, more stable. And if this is built in so that diversity is present in the building blocks, dynamics is present in the way they attach together, they can respond to changes in the environment. This is a chemistry which is dynamic in its constitution. In other words, the chemical object, be it a molecule or an assembly of molecules, can dissociate, fall into pieces, reassociate to remake an entity, and then this will then depend on what is present. And if something has happened during the dissociation and reassociation state, it will be able to respond to it. It will be able to adapt, leading to what you may call an adaptive chemistry. And that is now what we are mostly interested in, to try to design systems where adaptation is built into the system. In fact, maybe I can mention it. I don't have a slide for that. I have it somewhere, but I thought I would go over time. For the moment, I'm not yet over time. This is a case where the fact that in a given system, in the space of that system, a given temperature and pressure, if somewhere in that system there's a point which is more organized, then an entity of the exchanging pieces can go to that point. In other words, the possibility of getting order or getting an organized structure drives the formation of these pieces which make the structure. That's a sort of a catalysis of structure formation, and I really think that is one part of the development of complex matter under the thermodynamic physical laws of our universe. So you have understood perhaps that the essence of chemistry is not just to discover what is around. Of course, that's very important to discover what is around, but it can also create, and this is a strong word I am using, I'm perfectly conscious of that, novel expressions of complex matter, things which didn't exist before and which you can build. So the book of chemistry has to be written, not only to be read. And the music of chemistry, the score of chemistry, has to be composed, not only to be played. So chemistry can be considered as the art of matter. This is not so recent. Of course, it wasn't expressed in the same way as what I tell you, but Leonardo da Vinci, not a minor scientist either, not a minor engineer, a scientist, an engineer, and an artist, of course, has written this sentence, which I guess 100 years earlier would have been burned. Where nature finishes to produce its own species, that's what around us, what exists around us, man begins using natural things, the pieces, the periodic table of the elements, the bricks available in our visible matter in our universe, in harmony with its very nature. These are the laws of physics. I don't think Alain would contest that. To create, that's very strong, the ending. This guy was an artist, and not a minor artist. When he writes that, he knows what he's writing. To create an infinity of species, all those which don't exist yet, which you can make. Of course, the Lego set, the set of pieces is given. The periodic table of the elements in our universe is our set of bricks, and it's the set of bricks of the universe to make complex structures in our universe. So it's not just natural what we have to do, but man-made. And music is the same. It is also man-made. So, to finish with a touch of mathematics, not because I will give you any formulas, But a very famous mathematician, David Hilbert, he wanted on his tombstone in Göttingen to have 2 sentences written. The first one is a sort of a push, and this pushes the scientists. We must know. The second part is a vote of confidence. Wir werden wissen. We will know. I think if we wouldn't be convinced that we will know, then we would have less drive to try to know. Science shapes the future of humanity. And when I give this talk to high school students or so, I tell them, yes, participate. Not that I mean we should have only scientists. Mozart was not a scientist, and nevertheless the world without Mozart would be totally different. But if you want to be an artist, if you want to be a composer, you have to work at it, you have to train for that. Of course, for some people it's much easier. For Mozart it was very, very easy. It went automatically, so to say. It's self-organized. Oh, that's a nice case. Mozart had self-organized music in his brain, which in fact is true, because there's a score I saw in the museum of the Musikvereinigung in Vienna. I saw a page of Mozart where it was written, beim Kugeln komponiert, composed while he was playing, what is Kugeln? Sphere, balls, something like that. So that is quite interesting. He had it in his brain, he just prescribed it. So, music and science has another property, which is more and more and more important in our complicated world. Science has no borders. A law discovered on the planet Earth, a tiny speck of dust in an enormous universe, that law is valid everywhere. A molecule of water on our planet is the same on any other planet around the universe. That is why we can say that there is water on a given planet, because we know the properties of the water molecule. So we are, in some respects, it bothers me, we are limited to play with Lego set, which are the 92 elements. But we have still many possibilities, and we know that even from human beings on our planet here, the variety can be enormous. But the important thing is there are no borders. There's no border to music, there are no borders to science. And I think we better stick with that also on our planet, and especially also in Europe. Times to come will be shaky. I hope it will not be so shaky as to fall apart. We should all try not to let things fall apart. Thank you.
Jean-Marie Lehn.
Where are you?
Let's hope that we will not build the borders. We will keep walking together. Describe the world in 3024.
I can't.
Why not?
Not possible. Because, you know, these things in science, of course, there are things you can predict. For instance, physicists have laws, and if the theory is right, they can do. Gravitational waves had been predicted and have been found. It took a long time, and it was an enormous experiment where they detected a difference in length of less than the diameter of a proton. It's unbelievable. I almost think sometimes they are trying to make us believe in things which don't exist, but they did it, and it has been confirmed. So this can be done. But if you are a complex organism, like a living organism and so on, you have many interactions, many parameters. I can't tell you. And one example, a recent one, is the discovery of the vaccines. Because the vaccines have been, most people said, oh, that's a pandemic, we're in trouble for 8 years or 10 years. It was found in less than 1 year. Because the situation was more or less ripe. First of all, this also speaks in favor of diversity. Who made this vaccine? A Hungarian lady, Katalin Karikó, an American professor in Philadelphia. They recognized, in fact, it was a simple chemical change. This is something which people have forgotten because they got a Nobel Prize for Physiology and Medicine. But they did a lot of things. But the important thing, it can so be reduced to the fact that they took one letter of the alphabet I mentioned before, uridine, and turned it by 60 degrees, made pseudo-uridine. And that was enough to make it possible. Then, this was made practical by a company, not Pfizer, BioNTech in Germany. And this company was run by two people of Turkish ascendance, Ugur Sahin and Özlem Türeci. So, a Hungarian, an American, two people from Turkey. That's science. That's science. You don't know where the shot is coming from.
And sometimes just a slight change and the Nobel Prize is home. Jean-Marie Lehn, thank you very much.
Merci, monsieur.
Thank you so much.
Good to see you.
Great to see you. To be successful in science, it's necessary to be passionate about it, to find the right problem, to find the right advisor, to find the right solution. And it's always great when we have somebody who's passionate about something or someone. We have so many group of fans around the world. We have, for example, Trekkies, those who are fans of Star Trek, the made-up fictional characters. Then we have Swifties, those who are fans of Taylor Swift, the celebrity. And then we have those who are fans of the true celebrities, Einsteinians. One of the Einsteinians will come up on the stage in a moment's time. And he's not just an Einsteinian. He's the Nobel Prize laureate who helped to set a dispute between Albert Einstein and Niels Bohr. He was the one who helped to settle the dispute between these two scientific giants. He said, "My experiment was trying to find a limit of quantum mechanics, and we didn't find it." In the world of quantum mechanics, it's normal that you would be sitting here in this very theater and at the very same moment walking through a park with your dog. If you find it bizarre and weird, congratulations, you did the first step to understand quantum mechanics. Because our brains are originally not set to understand the world of quantum mechanics, despite it is all around us. In the film Love Actually, they said that love is all around us. But actually, it's science that is all around us. And in the world of quantum mechanics, there is a lot of potential, a lot of possibilities. And our next speaker will talk about the quantum revolution that can fundamentally change the world we are living in. Ladies and gentlemen, Alain Aspect. Professor, thank you very much for joining us.
I watch the watch. Yes, the hero of the two quantum revolution is definitely Albert Einstein. And I have a small book here, which is in French, but it's also in Italian, it's also in English. Maybe one day it will be in Czech, who knows. It's about Einstein and the quantum revolutions. And I am going to talk about the first quantum revolution, and you will see that Einstein is important. And I will talk about the second quantum revolution, and Einstein is yet more important. So let us start with there are two concepts for two quantum revolutions. The first concept is wave-particle duality, and the two heroes are Albert Einstein, who announced the idea of wave-particle duality for light in 1909, and my French fellow, Louis de Broglie, who announced wave-particle duality for matter in 1923. And then, at the root of the second quantum revolution, we have entanglement. And the hero of entanglement, again, is Einstein, with his colleagues Podolsky and Rosen, Schrödinger, who was also part of the first quantum revolution, and then another great scientist, John Bell. Let us start with the first quantum revolution. What means quantization? Why is this word quantization? Quantization is the fact that in modern physics, some quantities can only take some particular, we call it discrete, values, while in the classical world, this quantity can take any value. For instance, light. You describe it as a wave, and a wave expanding everywhere in the space can take any value for energy, for instance. But in the quantum world, as discovered by Planck and by Einstein, light may be considered from a certain point of view as made of elementary particles that we call photons, which have a definite amount of energy in them, so it's no longer continuous, is made of grains of energy. And of course, it's not because Einstein and Planck say that light is made of grain of energy that it was not a wave. Two great scientists at the beginning of the 19th century, my fellow citizen Augustin Fresnel, and on the other side of the channel, Thomas Young, these two great scientists have demonstrated that light must be considered as a wave. So, what to do with it? Well, Einstein told us a solution. In 1909, he said, light must be considered both a wave and a particle, and he gave many sophisticated reasons why you should consider both. Let us now come to quantization of matter and not quantization of light. So once again, some quantity can take only some particular values. And for instance, you have here the image of the atom with a nucleus which attracts electrons. And this is just like satellites going around the Earth or like planets going around the sun. The analogy is quite good. The force is exactly the same, goes as the inverse of the square of the distance. But there is a major difference. Satellite can be at any altitude, while in the atom, as shown by Niels Bohr, only some trajectories are allowed. Once again, it is quantization. And where does this quantization come? It comes from wave-particle duality. Now, for matter, electrons are elementary particles of matter, but you have to associate them with a wave. And you see the reason why you have here only some trajectories is the fact that if you consider the electron as a wave, the wave propagating in its trajectory, it has to come with an integer number of wavelengths, otherwise at the end of a full turn, it's a mess, it's not stable. And so it is this wave-particle duality that selects some specific orbits here. Okay? So the electronic orbits must be an integer number of wavelengths. So what do we do with this wave-particle duality? Well, first, we are happy. Look, Louis de Broglie and Edwin Schrödinger, they are really very happy that they have discovered wave-particle duality. Louis de Broglie discovered the relation between the wavelengths and the momentum of the particle, and Erwin Schrödinger does more. He discovers the equation describing these matter waves. But we can do better than to rejoice and be happy with wave-particle duality. In fact, wave-particle duality is at the root of our understanding of the world. Do you realize that by the end of the 19th century, physicists were quite sophisticated? They could not understand why matter was stable. What do I mean? Well, they knew that matter is made of positive and negative charges. and they knew that positive and negative charges attract each other. So why does not matter collapse? Normally, negative particle and positive particle should attract each other and matter would collapse. Why doesn't it collapse? Because from a certain point of view, you have to consider the electron as a wave, and as a wave, it cannot collapse. It has to have some wavelengths around the nucleus. So the first quantum revolution, wave-particle duality, the de Broglie relation, the Schrödinger equation allow us to describe the stability of matter, its mechanical properties, its electrical properties, and even properties that have been discovered later, well, not all of them, like superconductivity and superfluidity. But it's not only for the pleasure of scientists who are happy to understand matter. It also allowed the discovery of fantastic devices. I named them laser, transistor, integrated circuits. These devices were not discovered by a young guy in a garage in California. It was discovered by the greatest physicist of the time, trying to describe with quantum physics how electrons propagate in some strange material called semiconductor, or trying to describe precisely with quantum physics how light is emitted or absorbed with matter. And with that, they discover, or rather I should say invented, devices with the names laser, transistor, integrated circuit, which are at the base of our modern information and communication society. So the point that I wanted to make is that this first quantum revolution was first conceptual, with the concept of wave-particle duality, then scientific, allowing us to understand matter in its interaction with light, and then technological, and it has changed our society as much as the invention of the heat engine changed the society in the 19th century. Okay, let us go to the second quantum revolution. The concept at the root of the second quantum revolution is entanglement. I'm going to describe it with what is called a thought experiment, a Gedanken experiment. Einstein liked to make reasoning on Gedanken experiment. What is a thought experiment? It is an experiment which is possible according to the laws of physics, but that you cannot do because the technology available at the moment when you think of it, this technology is not available. That was very often the way of Einstein of thinking about physics. And he thought about something absolutely extraordinary, which is entanglement. Don't be afraid. When you go out of here, you will have learned something about Dirac notation. Dirac notation is this equation, and you are going to understand what it is about. But before describing that, I must tell what we have here. We have a photon going to the left, and you have the brother going to the right. So we have a pair of photons. Photon is light, and many of you know that light may be polarized. If you don't know, you have to think that something vibrates perpendicular to the direction of propagation. And in order to measure the direction of vibration, which is called the polarization, what you do, you use a device called a polarizer, which has an axis. And if the polarization is parallel to the axis, light will go up. And you say, oh, it's polarized along A. If it is polarized perpendicular to A, it goes down, and you say the polarization was perpendicular to A. But what happens when you have a beam which is polarized at an angle? A fraction of the beam goes up, and a fraction of the beam goes down. But hold on, I told you about quantization. If I have a single photon will not be split in two. So when you have a single photon, it will either go up or go down. And what quantum mechanics allows you to calculate is what are the probabilities to go up and down. Now let us come to the Dirac notation, and you will see at the end you have a lot of knowledge, okay? Let us put the two polarizer along the x direction, and this xx, we call it a bracket. The xx bracket describes a pair of photons polarized along x. If they arrive on the polarizer, both will go up. YY describes a pair of photons polarized along the y-axis. And if they arrive at the polarizer, both bow down. What Einstein and his colleagues Podolsky and Rosen discovered is that the quantum formalism allows you to think not only of XX or not only of YY, but also on the sum. And so what are you going to tell me? And so what? The point is now this sum, which is an entangled state, it is impossible to describe the pair of photons by saying, oh, this photon is like that and the other photon is like that. No, it's a global description, and it's impossible to go out of this global description. In a sense, it's as surprising as the Schrödinger cat, which is both dead and alive. Here, my pair of photons is both XX and YY. Surprising, I can tell you. What can we do with that? Well, if you follow an elementary course in quantum physics, you can calculate the probabilities to get the photon in channel +1 or -1. And so the first thing you calculate are the single probability, and you find P plus equal P minus equal 1/2, which means that for this photon, whatever the orientation of the polarizer, you get 50% chance to get +1 and 50% chance to get -1. Same for the other photon. So you see, it looks totally random. You have something which is totally random here and something which is totally random there. But quantum mechanics also allows you to calculate joint probabilities. The probability to get +1 here and +1 here, or the probability to get -1 here and -1, or the probability to get +1 and -1, etc. And the result of the calculation, when you have this entangled state discovered by Einstein, Podolsky, and Rosen, the result is really interesting. It tells you that if you get +1 on that state, you get +1 on the other side. And if you get -1 here, you get -1 here, and never the cross results. This is surprising. We call it, in science, a strong correlation. There is perfect correlation between two random events. But I like to call it also a tricky coin toss. You know, think, you have a guy here throwing a coins. He finds head or tails. You have here another one throwing your coins. He finds head or tails. And when you look separately, it looks totally random. But you, in the room, with some recoil, you observe the two, and you say, this is strange. If this one get heads, this one get heads. If this one get tails, this one get tails. There is a trick. You want to know the trick. You mentioned that I like to do card tricks. But when you have a magician doing card tricks, it's not magic. There is a trick below. You want to understand. And I want to understand what happens here. Okay, what can we do? Can we find an image corresponding to the calculation? Calculation tells us that there is a strong correlation. Can we find an image by looking in detail at the mathematics? Well, the result is both disappointing, but we also learn something from it. The calculation is done globally on the pairs of photons, and then you get the result of the calculation. But this calculation happens in an abstract space called the Hilbert space. We heard of Hilbert already today. And this calculation done in an abstract space allows us to visualize, in a sense, what happens. But I'm not happy with an image in an abstract space. I live in an ordinary space. My laboratory is an ordinary space. I have a source in the middle, a polarizer here, a polarizer there. So I would like to, and so here you see the problem is that one cannot identify individual property for each photon. But as a famous theorist in quantum optics say, quantum phenomena do not occur in a Hilbert space. They occur in a laboratory, and I would like to see what happens in a real space. I'm not going to do it in details, but I can tell you that there is another way of doing the calculation which gives us a description, an image in our everyday space, in our ordinary space. And the image is the following. The image is, according to quantum mechanics, until the very last moment, each photon can give the result +1 or -1. But at the very moment when photon U1 takes the choice +1, then instantaneously the other photon takes the channel +1. And if the first photon takes the channel -1, then the second one takes the channel -1. So you know, this image is quite crazy, and Einstein could not accept it because the measurement on the first particle seems to influence instantaneously at a distance the state of the other one. We call it quantum non-locality, but it seems to contradict relativity, according to relativity of Einstein. Nothing can go faster than light. Okay. So this was absolutely unacceptable for Einstein. But Einstein came with a different reasoning, and I'm sure that many of you already thought of it. The reasoning is the following. Let us assume that from the beginning, the two photons have a joint property that we call lambda, the same property. Maybe both have an X polarization, so both will go up. And for the next pair, maybe both have a Y polarization, both will go down. And with such an image, you can understand the correlation. And I want to emphasize that this image is not only simple and convincing, but it's very scientific. It's analogous to the conclusion of medical doctors or biologists when they observe that there is a common property between twin brothers. They conclude that this common property is due to the fact that they share the same chromosomes, the same genes. So, you know, this is a very nice idea, and it was a conclusion of Einstein. You have to complete quantum mechanics introducing these supplementary quantities, these supplementary parameters. Niels Bohr disagreed. Niels Bohr has a strong intuition that you cannot complete, you cannot add anything to quantum physics. It's only XX plus YY, period. Don't add anything to it. It was a long-lasting debate, never settled. I mean, they died, Bohr in 62, Einstein in 55, and were arguing all their life about that. And this debate between two giants drew absolutely no attention by the young physicists. You know, young people do not care for what old people say. Quantum mechanics was accumulating success. And moreover, the discussion was about interpretation. Einstein did not disagree at all with the way to do the calculation in standard quantum mechanics. He just wanted to add something to the interpretation. But he did not say that there was a difference between his point of view and the point of view of Bohr regarding the result of the prediction of the calculation. And this is a good reason why young physicists did not care. They did not care until the moment when a theorist at CERN in Geneva discovered that if you follow the point of view of Einstein, then at one point you come to a prediction, to a result of a calculation, which is different from the result of the calculation of standard quantum mechanics. Which means that he pointed out a quantitative disagreement between the points of view of Einstein and Bohr. And so, to my knowledge, for the first time, you could settle a debate between philosophical point of view, you could settle it with an experiment. What? C'est ici. In fact, there were 3 generations of experiments. The third generation was done in Berkeley, in Harvard, in 72, and there also at Fry in Texas. John Clauser, which was one of the recipients of the Nobel Prize in 22, found a result in agreement with quantum mechanics, while Pipkin in Harvard found a result contradicting quantum mechanics. But there was some kind of a trend in favor of quantum mechanics. In fact, there were 4 experiments, and the score was 3-1. Anyway, all these experiments were very far from the schemes that I have described up to now, and this is why I embarked in 74, 75 in a new series of experiments, the goal being to have experiment really very close to the thought experiment, to the Gedanken experiment. And in particular, in one of these experiments, I could demonstrate that, yes, until the last moment, it can still be +1 and -1, and it is only at the very last moment that the decision is made. And there was a third generation of experiments starting, so my experiments were published in 82, and there was a new series of experiments starting in the late 80s and culminating in 2015, where several groups announced loophole-free test of Bell's inequality, loophole-free meaning there is no possibility to argue that there is something which is not perfect in the experiment. Okay. What is the conclusion? The conclusion is that we have to abandon the vision of Einstein of the microscopic world. This vision is called local realism. Local realism is the fact that when I have 2 systems, for each system I can use quantity to describe what he calls the physical reality. That is to say, quantity which allow us to calculate the result of any measurement we do on this one. And there is another one here, and it's the same for it. And there is no possibility for these 2 entities, for these 2 objects, to interact instantaneously. Well, we have to abandon this point of view. And we have to admit, according to Einstein himself, told us, if I was not right, this is what you would have to accept. But now we know that he is not right, so we have to accept one of the 2 things that he said. The first thing is abandon the independence of the physical reality present in different objects, in different volume of space-time. We call it quantum holism, which means these 2 entities still form a whole. The other point of view is to accept an instantaneous interaction between the 2 objects. This is called quantum non-locality. And why is it nice to have these 2 visions, which have been designed by Einstein himself? It's because quantum holism leads to application in quantum computing, and quantum non-locality leads to application in quantum communication. And I will just show you in the last minute that I have. But everybody goes beyond the time, so there is no reason that I cannot do it. I will show you how the concept of quantum non-locality leads to a fascinating quantum technology, which is called quantum cryptography. This is what it is about. You have 2 friends. Alice, you recognize her, okay? And Bob, you recognize him, okay? And they want to exchange a message, and there is the spy, the eavesdropper here, which would like to intercept the message, okay? And there is a solution to have an absolute secret communication. It was designed by Robert Shannon, the father of information theory. And Shannon said, if Alice and Bob have 2 identical series of random 1 and 0, Alice uses this K for encoding the message, Bob uses this K for decoding the message, and the secret is absolutely guaranteed under one condition. The message must not be longer than the key, because otherwise there is a kind of periodicity, and the people who try to decipher are very smart mathematicians, and they will discover something. So the problem boils down to the fact that you want to distribute to Alice and Bob 2 identical series of 1 and 0. And quantum K distribution does the job. If you remember what I told you, if I have here a source of entangled photon, I send one to the left and one to the right, and Alice and Bob change their orientation, but from time to time, Alice and Bob take the same orientation for their polarizer, and at that moment, if Alice gets +1, Bob gets +1. If Alice gets -1, let us say 0, Bob gets the same result. Why is it interesting to know that this happens at the last moment, that you have non-locality? It's because, look, for the spy we see here, there is nothing to spy, because until the very last moment, it can be as well +1 as -1. And once you realize that non-locality tells you that there is nothing to spy, you have understood how smart is that method invented by Arthur Eckert. Of course, there is a lot of theoretical treatment, etc., but it's enough to think in terms of non-locality to know that it is a good idea. Let us go. Just to mention, our Chinese friends have demonstrated quantum cryptography with entangled photon over more than 1,000 kilometers by having a satellite emitting entangled photons. It's time to go to the end. The second counter-revolution is based on entanglement. You have understood, I already explained, and also on the fact that starting in 1970s, physicists have learned to manipulate and observe single quantum objects, single electron, atoms, and here my friend and former student, Antoine Broes, is able to manipulate individual atoms. Here, just to play, he shows an Eiffel Tower, but in fact what he does, he arranges several hundreds of atoms to build a quantum computer. Okay, so to conclude, we have quantum technology derived from the basic concept of the second quantum revolution, entanglement and manipulation and control of single quantum objects. And these technologies are quantum communication, quantum computing, quantum metrology. Just to impress you, this gravimeter measures gravity with a method based on quantum physics. And this works so well that you can put it on a volcano to observe small fluctuation of gravitation in the hope that you can see precursor of volcanic eruption. So there is one on the Etna volcano, for instance. Quantum computer, we're in the process where quantum computers are at the stage where they may well be overcoming classical computer. It's not yet settled. We don't know. It's an open situation. This is why science is so interesting. And quantum communication, I told you already, quantum cryptography, it works. So, will we have a second quantum revolution? Will it have a major impact on society? You remember the three criteria? A concept, entanglement, yes. Scientific application, yes. Will it change the society? We don't know yet. There is a famous quantum advantage, and to finish, I would like just to emphasize that quantum advantage can be two different things. Either a computing power bigger than for classical computer, or doing the same kind of calculation, but saving energy. And with this, nobody can answer with certainty if it will change the society or not, but many believe in it. Just to give an example here, there are programs with billions from Europe, France, Région Île-de-France, that is to say around Paris, the region Île-de-France, and many startup companies will play the game. This is fascinating perspective. It's really science. We don't know yet what will happen out of that, but it is an exciting time. Thank you very much. Now he's going to torture me.
Pardon?
Torture.
Torture?
Torture.
I understand that you said he will be talking germs. No, but quantum computing.
Yes.
In which areas, in which areas of scientific research can it be truly helpful? Could you point a few?
Many. For instance, describing big molecules, the kind of molecules that can be used as drugs. Normally, quantum computer can do it better than classical computer. AI, we spoke of artificial intelligence. Sometimes you know that artificial intelligence must be trained on a corpus of things. But sometimes you don't have enough data. The quantum computer can generate data to train artificial intelligence because there is a big difference between quantum computing and artificial intelligence. Artificial intelligence is a black box. You feed data, and then it gives an answer, and nobody understands how it works inside. On the other way, the quantum computer, you know exactly what it is doing. So you can generate data of which you are quite certain, feed it to the black box, and hopefully it helps artificial intelligence. Isn't that beautiful?
Alain Aspect. Thank you very much, Professor.
Thank you.